Assalamu alaikum. Hi everyone, welcome to my lecture. It will be about apoptosis and cell death. In the next coming slides, I will explain key concepts in apoptosis and cell death. Okay, what is programmed cell death, or so-called apoptosis? In our body, if cells are no longer needed, they commit suicide by activating an intracellular death program. So it is a program that removes the unwanted cells or kills the unwanted cells. This process is therefore called programmed cell death. Okay. Apoptosis, from the Greek word, means falling off as leaves from the tree. Why apoptosis? Or, in other words, what are the functions of apoptosis? Or what functions does apoptosis serve? Okay, we have few examples here. The first example is during development. And during development, we have this frog here. During the development of frog, for example, this called a tadpole. And this is a frog. In order for a tadpole to transform into a frog, it has to lose its tail. That means all the cells in the tail, they supposed to die, and this happens via apoptosis. So apoptosis to all the cells in this tail to lose it, and then the tadpole becomes a frog. Another example is what happened in cell death that balances cell division, or balancing between cell death and cell division. Okay, let's have an example, the liver. If part of the liver is removed in an adult rat, for example, liver cell proliferation increases to make up the loss. So we have cell division to compensate for the loss. And conversely, if a rat is treated with a drug, phenobarbital, and phenobarbital is a drug which stimulates liver cell division, what will happen? Therefore, there will be a liver enlargement. Okay? What will happen if we stop this drug? If we stopped phenobarbital, apoptosis in the liver greatly increases until the liver has returned to its original size, usually within a week or so. So, the liver is kept at a constant size through the regulation of both the cell death rate and the cell birth rate. And by the cell death rate, I mean apoptosis, regulation between apoptosis and the cell division. Another example is when cells are damaged or stressed. For example, DNA damage will induce, or in DNA damage, p53, tumor suppressor gene, will activate a pathway for apoptosis, and we will see this later in the upcoming slides. Okay, here in this table are some of a key comparison points between apoptosis and necrosis. As you know, both of them are cell death, but what are the main differences between them? Okay, for me, the main difference that I want you to know in this I want you to know in this slide is the inflammation. That there is no inflammation in apoptosis, and there is inflammation or inflammatory response in necrosis. Okay, let's review the points one by one. Regarding regulation, as we said, apoptosis is highly regulated and planned. On the other spectrum, necrosis is accidental and not regulated. Okay, regarding the cell, in apoptosis there is shrinkage to the cell, while in necrosis there is swelling and cell lysis. And we came to the plasma membrane. There is what's so-called blebbing of the cell membrane. Blebbing means forming a blister, like the blister that is formed in burn injury. This is blebbing. So there is blebbing to the plasma membrane in apoptosis, but with intact integrity. So there is no spilling over to the contents outside the cell. Okay, and there is formation to apoptotic bodies at late stages. While in necrosis, there is loss of integrity and increased permeability. And as you might imagine, there is spilling over to the contents of the cell outside the cell. Okay, regarding the nucleus, there is chromatin condensation in both of them. Here there is also fermentation, and here there is disintegration or degradation of the nucleus. Okay, the two mechanisms are apoptosis and necrosis. Which one of them requires energy? Think of it. Yes, it is apoptosis. Apoptosis requires energy, ATP dependent, while necrosis is ATP independent. And here we said, here there is no inflammation, and the dead cell is cleared by phagocytosis, usually from a neighboring cell. So it's a neat process, and it causes no harm to the surrounding cell. It is very important to know this. While in necrosis, there is inflammatory response with damage to the neighboring cell. We said that, for example, if the cause of necrosis was to be a pathogen, for example, bacteria, this bacteria, if there is necrosis and spilling over the contents outside the cell, this bacteria might infect the surrounding cells and cause damage to them. Okay, apoptosis is mediated by enzymes, and the group of enzymes that mediates apoptosis are caspases. They are called caspases. Caspases are proteases enzymes. Okay, they are present in all nucleated cells in form of inactive proteases. Called procaspases. So procaspases are the inactive form of caspases. These procaspases that lie waiting for a signal to destroy the cell. Okay? Therefore, it is not surprising that caspase activity is tightly regulated inside the cell. Okay, the pathways of apoptosis. There are two pathways of apoptosis, intrinsic and extrinsic pathway. And intrinsic, from its name, means from inside the cell, while extrinsic means from outside the cell. But both end up in aggregating procaspases together in a complex, and then their activation. There are details here, but we are not going to go into details. Just know that there is aggregation to the procaspases in a form of complex, and then after their aggregation in a complex, that will be their activation. So aggregation is the first step in their activation. Okay, the first pathway, intrinsic pathway, we said that intrinsic means from the activation here is from within the cell itself. Here we have the mitochondria. The mitochondria is induced to release the electron carrier cytochrome C. Okay, cytochrome C, released from the mitochondria to the cytosol. Cytochrome C activates adaptor protein, which then activates procaspases. So this is how cytochrome C activates procaspases. It first activates adaptor protein, and then this adaptor protein activates procaspases. Example for this in DNA damage, we have, as we said previously, p53. p53 indirectly promotes the release of cytochrome C from the mitochondria, and then therefore activation of intrinsic or mitochondrial pathway of apoptosis. Therefore, we say that p53 is apoptosis promoter or apoptosis inhibitor. p53 will be apoptosis promoter, right? Okay, the extrinsic pathway. The extrinsic pathway, extrinsic means we said from outside the cell, and the activation here of death receptor protein Fas, or death receptor Fas protein on the cell surface. This receptor activated by a ligand called Fas ligand. Okay, the binding of Fas ligand to Fas death receptor on the cell surface of a cell that will undergo apoptosis. This will activate the extrinsic pathway of apoptosis. So it's a receptor ligand interaction. Which receptor? Death receptor protein Fas, or Fas protein death receptor. And which ligand? Fas ligand. Okay. From where do we get Fas ligand? Fas ligand can be produced by killer lymphocytes, but sometimes it can be produced from within the stressed cell to kill itself. Okay, in some occasions. Okay, we said that apoptosis is highly regulated, and regulation means sometimes inhibiting it, sometimes activating it, okay, or sometimes promoting it. So what regulates apoptosis? There are two families of proteins. The first family, PCL2 family of proteins. In this family there is apoptosis inhibitors as well as apoptosis promoters. Example for apoptosis inhibitors from this family: PCL2 itself, PCL2, and PCLXL. And example for apoptosis promoters are BAX and BAC and BID. Okay. The other family is IAP family of proteins, inhibitor of apoptosis. Okay. As we might imagine, if there is something wrong with the regulation of apoptosis, this can yield either excessive or insufficient apoptosis, either too much apoptosis or too little apoptosis. Will that be a problem? Of course, yes. Both can contribute to a disease. Both too much apoptosis and too little apoptosis can contribute to the disease. Excessive apoptosis, for example, in brain and heart cells that die after reperfusion in strokes and heart attacks. To explain this, let's take stroke for example. In a stroke, the first or the initial injury and cell death occurs due to ischemia and loss of blood supply. But after some of the cells die due to ischemia and decreased blood supply. But after reperfusing the area, for example by IV thrombolysis, another group of cells die, and then they die by apoptosis. And this, as you might imagine, this exacerbates the damage caused by stroke. Another the examples for insufficient apoptosis are mutations in p53 tumor suppressor gene. We said that p53 has apoptosis promoter activity, right? So it promotes apoptosis. So as you might imagine, mutations in p53 tumor suppressor gene will cause insufficient apoptosis or decreased apoptosis, and this leads to cancer cell formation, right? Actually, p53, there are 50% of mutations in p53 in human cancers, okay? Or to say this in another word, 50% of human cancers, there is p53 mutation within them, okay? Another example for insufficient apoptosis, mutations in either fast death receptor or its ligand. And of course, this will disable the process of apoptosis, yielding in too little apoptosis, because fast death receptor and fast ligand both are involved in apoptosis in the extrinsic pathway, certainly. This is the end of my slides. I hope I was able to clarify important or key concepts in apoptosis, and thank you.